Okay, so I guess we can start on our lecture today. Okay. So on our last lecture, we stopped at this slide, okay, where we looked at the characteristics of adaptive immune response. Okay, adaptive immune response. So this, just to recap, okay, for the adaptive immune response, as you know, it is the third line defense where your body will protect against the invasion of specific pathogen. Okay, specific pathogen. So for the first characteristics, as you can see here, the response is rapid to second exposure to the same pathogen. Okay, so because the adaptive immune response, it triggers or it protects your body against specific pathogen. So uh, in the process, okay, in the process of protecting your body against that specific pathogen, your body will produce memory cell. Okay, so this memory cell will it has a long lifespan where it will protect your body against the same pathogen later in your life if you are exposed to the same pathogen again. Okay, so due to the presence of the memory cell, for example, memory B cell, once the pathogen entered again in your body, the same pathogen entered again in your body, the memory cell can be activated and then straight away can produce antibody. Okay, can produce antibody that circulate in your body fluid to protect against that specific pathogen. Okay, so and then okay, for the second characteristics, okay, it detects antigen never before encountered. Okay, so because the adaptive immune response, it involves um, lymphocytes, okay, B cells or T cells, okay, B lymphocyte or T lymphocyte. So in your body there are many B cells and T cells that have receptors, okay, receptors. So the receptor has antigen binding site, and this and the receptor of the lymphocyte will bind to any antigen, okay, any antigen that has many different shapes. So the binding of the receptor can activate the adaptive immune response. So any pathogen that brings antigen into your body, okay, will be detected, okay, will be detected by the receptor of the B and T cell. Okay, so you have many B cell and T cell with different receptors, and this will be detected. This will be used to detect the antigen. Okay, the antigen, which is the foreign molecules that enter into your body, such as the molecule or the antigen on the pathogen. Okay. Okay, so then the next characteristics, number 3, able to distinguish self from non-self antigen. Okay, self antigen, okay, and non-self antigen. So self antigen are molecules that are found in your body, your cell body. Okay, so that is self antigen. So your immune system should not attack self antigen. Okay, so your immune system will not attack self antigen. If your immune cell attacks self antigen, so it will trigger a disease. We call it as autoimmune disease. Okay, autoimmune disease. And then kalau non-self is the foreign stuff. Okay, that is foreign from pollen or from antigen, from other molecules that enters into your body. So this one is protected by your immune cell. Okay, so your immune system supposed to attack non-self antigen. And then the fourth characteristics is cell proliferation. Okay, so what is cell proliferation? The specific B cell or T cell that detects that specific antigen will be activated and divide many times, okay, through the process of mitosis. Okay, so that your body produce the same B cell with the same receptor to protect or to attack the same pathogen. Okay, proliferate into many cells. Okay, proliferate, divide into many cells. So increases the number of B and T cell specific for the antigen. So these are the 4 characteristics of adaptive immunity that you have to remember. Okay. So saya harap tadi Aisyah Nadira dah masuk. Okay. Tapi nama dia ada Nur. So okay. Alright. So next, cell involved in adaptive immune responses. So there are two key cells, two key cell types, which is lymphocytes and also antigen presenting cell. So lymphocyte, we have two groups, which is the T lymphocyte or T cell, okay, and then the other one is the B lymphocyte, okay, or the B cell. Alright, so the antigen presenting cell is the cell that will engulf the pentogen. So there are three different types of cells that will become antigen presenting cell, or we call it as APC. So one is macrophage. Okay, second is B cell. B cell can also act as APC. Okay, and then the other one is dendritic cell. Okay, so these are the 3 cells that will become APC, and these 3 cells are capable, they are going to engulf the pentogen, degrade or destroy the pentogen into fragments, into antigen fragments, and then the antigen fragment that is produced by these 3 cells will be presented on the surface of the cell. Okay, for example, here you have a macrophage. So the macrophage will engulf a pentogen, okay, and then engulf and then degrade the pentogen into fragments, antigen fragment, and then there will be a type of protein, we call it as MHC molecule, okay, a major histocompatibility protein, which is a protein produced by the macrophage or B cell or dendritic cell. So these, you have to remember, these are the host cell dalam badan awak lah, host kan. Okay, host cell. Okay, so the MHC will bind to the antigen fragments, okay, and then the antigen fragment will be presented on the surface of the APC. So that's why we call it as antigen presenting cell. Sel ni akan engulf, degrade, dan dia akan presentkan pada permukaan surface cell ni. Okay, so dia nak presentkan kepada siapa? Dia nak presentkan kepada sel yang kita namakan dia sebagai T helper cell. Okay, T helper cell. So nanti kita akan tengok. Later we are going to look at the mechanism of this reaction. Okay. So the interaction between the APC and also the helper T cell. Alright. Okay. So the T, it says here, point number 3, T cells and B cells recognize antigen. So all lymphocytes develop from stem cell in the bone marrow. So basically, the bone marrow is where T cells are produced. Okay, so the B cell complete development in adult bone marrow. So B cell, it stays in the bone marrow to mature. Okay, and then as for the T cell, it will migrate to the thymus to mature in the thymus gland. Okay, so that is the place where the T cell will mature in the thymus. B cell matures in the bone marrow. Okay, so this lymphocyte will roam in tissue and organ of lymphatic system to induce immune response. So lymphatic system, so it can travels to the lymph node, for example, lymph nodes. Okay, lymph nodes. So basically, lymph nodes is the organ of the lymphatic system. It is where memory cell will be stored. Okay, so memory cell will be stored in the lymph nodes. So binds to antigen and undergo clonal selection to produce clone of cell. So Clonal selection, as I said, specific B cell with specific receptor against specific pathogen will be activated and then divide many times, okay, undergo proliferation. So that is clonal selection. So it divides many times, producing B cell with the same receptor or T cell with the same receptor, and then this B and T cell will then differentiate into two groups or into two clones. We call it as effector cell, okay, effector, or the activated cell that will act during the time of infection, and the other one is the memory cell. Okay, so this applies for both B and T cell. Okay, so T cell dia ada dua jenis. Okay, we have the T helper cell, and the other one we have the T cytotoxic cell. Okay, so this one you have to remember. Okay, so the effector B cell, kalau B cell effector dia, we call it as the plasma cell. Okay, so this plasma cell is the cell that is responsible for producing and secreting antibody. Okay, antibody, right? So the antibody will circulate in your body fluids. Okay. Okay, so this table basically it shows the difference between B and T cells. So B cells, it matures in the bone marrow, kan? So nak ingat dia, the B cell mature in the bone marrow, and T cell matures in the thymus. Okay, so and then B cell secretes antibody. Okay, so the effector, I will write it again. The effector B cell that is responsible for secreting the antibody, or we can also call it as the plasma cell. Okay, and then as for the T cell, directly attack the cell infected with pathogen. This one is referring to the cytotoxic T cell. Okay, directly attacks infected body cells. Okay, body cells or the whole cell that is infected with the pathogen. So this cell will be destroyed by the cytotoxic T cell. Okay, so for the B cell, it mounts or activate the antibody mediated immunity, or we can call it as the humoral immune response. Okay, humoral immune response. As for the T cell, it mounts or activates the cell mediated immunity or immune response. Okay, so for T cell, as I've mentioned, there are two types, which is the T cytotoxic T cell, or you can abbreviate as TC, that TC again. So subscript C. So the T cytotoxic cell will destroy cells infected with Pentogen. Okay, cells infected with Pentogen. Cell is referring to the host cell. Okay, and then the helper T cell or TH, which is cells that activate B cells and cytotoxic cells by secreting cytokines. So basically, the helper T cell is the key cell. Okay, it is the most important cell, because the helper T cell is the cell that will produce a signaling molecule. We call it as cytokines. Okay, so the cytokines produced by the helper T cell will be used to activate the B cell and also the cytotoxic T cell. Okay, so without the helper T cell, kalau tak ada helper T cell, or if your T cell is being destroyed, for example, or you are lacking helper T cell, so your body cannot activate the antibody mediated immune response and cannot activate the cell mediated immune response. Okay, ini yang paling penting ni, the helper T cell. So later on when we looked at diseases related to immune system, so you will find that if a person has AIDS, okay, the virus, which is HIV, will destroy the helper T cell. So that's why if a person has AIDS, that person, the patient or that individual, will have a very low immunity. Okay, they have a very low immunity. So because the helper T cell is being destroyed by the virus. Okay. So next, okay, so in this diagram here, you see that the difference between the antibody mediated immunity and also cell mediated immunity. So antibody mediated immunity, it involves the B cell. Okay, so the cell mediated immunity, it involves the T cell. Okay, so the T cell matures in the thymus, the B cell matures in the bone marrow. Okay, so as you can see at the surface, okay, at the surface of these two lymphocyte, you will see that there's receptor. Okay, receptor. So this receptor here, okay, will bind to specific antigen. Okay, they will bind to specific antigen, okay, of a Pentogen, okay, of a Pentogen, right? Of a Pentogen. So but okay, for one specific B cell or for one specific T cell, all the receptors, okay, all these receptors will bind to the same antigen. Okay, they will bind to the same antigen. So this is what we call it as the antigen binding site. Okay, antigen binding site on the receptor of B or T cell. So B cell, T cell ni dia akan ada banyak reseptor, tapi reseptor dia tu akan bind kepada antigen yang sama. Sebab reseptor dia shape dia sama je. Okay, cuma kenapa dia ada banyak, to increase the probability to bind to that specific antigen. Bayangkan kalau ada satu je, kalau molekul antigen tu ada belah sini, macam mana dia nak bind, faham? Okay, so that's why the B cell has to have many receptors. Okay, many receptors, but the receptor are the same for the same B or T cell. Okay, so sama juga ini kita the receptor has antigen binding site. Okay, specific to bind to specific antigen. Okay, so and then the B and T cell will then it travels in your lymphatic system, and it can be stored in the lymph nodes or the spleen or other lymphatic organs. Okay, final maturation of B and T cells in the lymphatic organs. Okay, so immune system ni dia relate juga dengan lymphatic system lah. Okay, so next is the T cell receptor and B cell receptor. Okay, so B and T cell has receptor on the surface of the plasma membrane of the lymphocyte. So the receptor molecule are actually proteins. Okay, the receptor molecules are actually proteins. Okay, so protein that is bounded to the membrane of B and T cells respectively. So short form kita boleh panggil juga, kalau untuk T cell tu adalah TCR, T cell receptor. Okay, T cell receptor or TCR binds to antigen fragment that is displayed or presented on the surface of host cell by major histocompatibility complex. So MHC. Okay, MHC is a protein molecule produced by the host cell. Okay, by the host cell. So we have two types of MHC, MHC class 1 and also MHC class 2. Okay, MHC class 2 is produced by the cell that becomes APC. Okay, the cell that becomes APC, apa tadi? Macrophage, B cell and also dendritic cell. Okay, MHC class 1 is produced by infected body cell that has nucleus. Okay, infected body cell that has nucleus. So these are basically host cell. So when Pentogen is inside the host cell, so if the host cell is macrophage, the macrophage will degrade the Pentogen into fragments or antigen. So the antigen will bind to this protein Okay, will bind to this protein which is the MHC and presented kepada siapa? Presented or displayed. Okay, it is displayed to the T cell. Okay, it is displayed to the T cell. Okay, it is displayed to the T cell. So kalau APC dia displayed kepada helper T cell. Kalau nucleated cell, it displays to the cytotoxic T cell. Okay. So for the B cell receptor, it binds to antigenic determinant or epitope. Okay, so you have to know this term, epitope. Okay, of an intact antigen on Pentogen or circulating freely in body fluid. So for example, you have a virus. For example, on the surface of a virus has the antigen molecule. Okay, has the antigen molecule. Alright, and then this antigen molecule, it has a small structure, a very small structure. So this specific structure, or we can call it as epitope. Okay, epitope. So this is where actually the sinilah kot okay, macam tu saya buatkan untuk semua lah. Alright. So this is where actually the receptor of the B cell will bind, for example. Okay, so this is receptor of B cell will bind. Okay, so that is epitope. Okay, B cell receptor binds to antigenic determinant or epitope of intact antigen on Pentogen or circulating freely in body fluid. Circulating freely in body fluid. Okay, so the virus can be found in your body fluid. What are example of body fluids? Is your blood. So if the virus is in your blood, or the virus is in your interstitial fluid, or the virus is in your lymph. Okay, so they can circulate anywhere in your body. Okay, anywhere in your body, and then in your body fluid also there are antibody. Okay, antibodies. Okay, antibody. So the circulating antibody will have the same antigen binding site as the B cell, because the B cell is secreting and is producing and secreting the antibody. So obviously dia akan detect epitope yang sama lah. Okay, so the antibody will also bind to the epitope circulating in your body fluid. Okay, blood, lymph, and also interstitial fluid. Okay, so this is the structure of B cell receptor and also T cell receptor. So kalau yang bentuk Y ni adalah B cell receptor. Okay, bentuk Y ni adalah B cell receptor, bentuk Y ni kan. Okay, tapi kalau yang T cell receptor dia straight je. Okay, straight macam ni. So we look at the B cell receptor first. So for the B cell receptor, if you can see here, it says here, the B cell receptor has two identical heavy chain. This is the heavy chain. Satu. And then the other one satu. Okay. Two identical heavy chain and two identical light chain. Light chain yang pendek sikit ni. Okay. Two identical heavy chain and two identical light chain linked by several disulfide bridge. So they are linked by disulfide bridge. Kena lekatkan. So the attachment is done by the disulfide bridge. Okay, so kita belajar dulu kan disulfide bridge masa semester 1. Alright. Okay, so you have to remember that the receptors are protein molecule. So because they are protein molecule, the protein are made up of what? Polymers of amino acid. Okay, polymers of amino acid or polypeptide chain. So what is V, what is C here? Okay, V dengan C. V dengan C ni apa? Okay, kalau V ni adalah variable region. Kalau C ni adalah constant region. Apa yang constant, apa yang variable? The arrangement or the sequence of the amino acid. Okay, the arrangement or the sequence of the amino acid of the receptors. Okay, so receptor ni adalah protein. Protein diperbuat daripada susunan amino acid sequence. Okay, jujukan amino acid. Okay, so the amino acid sequence, kalau kat bahagian C ni constant, it means that your body has many B cell. Okay, B cell yang saya tulis hari tu B cell 1, B cell 2, B cell 3, B cell 4 sampai seterusnya. So the sequence of the amino acid will be the same from one B cell to another B cell. That is the constant region. Constant region amino acid dia sama. B cell 1, B cell 2, B cell 3, B cell 4, 5 seterusnya. The constant region will have the same amino acid sequence. What makes the B cell different is at the antigen binding site, which is the variable region. So there, the amino acid sequence will be different. That is the variable region. The sequence of the amino acid will be different for between B cell 1, B cell 2. Sebab apa? It determines the antigen binding site. Okay, because this is where different antigen or different epitope will bind to. Okay. And then for the receptor, it has the transmembrane region. So this is the region that penetrates through the plasma membrane of the B cell. Okay, region yang lalu plasma membrane tu we call it as the transmembrane region. Okay, so this is the B cell. So same goes to the T cell. Cuma T cell dia ada 2 chain sahaja. Okay, 2 chains, which is the alpha chain. One alpha chain and one beta chain. Sama juga dia ada constant region. So it means that from one T cell to another T cell, the amino acid sequence will be the same, but the variable region will be different. The sequence of amino acid for the variable region from one T cell to another T cell will be different because it determines the antigen binding site. So different T cell will bind to different antigen. Okay, so this is determined by the variable region due to the different sequence of amino acid. Okay, at the variable region. Okay, so now we are going to look at the definition for Pentogen. So what is Pentogen? It is an organism or virus that will cause disease. Okay, so any virus or any bacteria or any multicellular pathogens, okay, so they are foreign, they are outside of your body. So when it enters into your body, it can cause you to get certain disease. Okay, so antigen, what is antigen? Which is a substance that elicits immune response or trigger an immune response, okay, or stimulate an immune response. Okay, so has several different antigenic determinants, epitope tadilah, okay, on its surface, induce an immune response recognized by T cell and B cell receptors. Okay, so you have a Pentogen, okay, you have the antigen on its surface and an epitope macam tu, kan? So macam tu lah, kan? So the epitope is where the receptor of B cell, kat sini yang dia boleh be a receptor of B cell ataupun T cell or even antibody. Tulis kat sini lah, antibody will bind to. Okay, so receptor of B cell will have the same shape as the antibody. Okay, receptor of B cell will have the same shape as the antibody. So maksudnya antigen binding site of the B cell receptor and also the antibody, it is the same. Okay, the same. Cumanya kalau antibody dia tak ada transmembrane region lah, sebab the antibody is secreted into your body fluid. So the antibody is lacking the transmembrane region. So B cell receptor, yes, saya repeat the B cell receptor has the same shape as the antibody secreted by the plasma cell or the effector B cell. Okay, same shape, except that it is lacking the transmembrane region because the antibody is secreted. Okay, secreted into your body fluid. So the binding of antigen to B cell receptor initiate the production of antibodies from plasma cell. Plasma cell is effector B cell. Okay, effector B cell. B cell that is going to be activated and work at the time of infection, okay, to produce antibodies. Okay, so more on antigen. Okay, so antigen is a large molecule, either proteins or polysaccharides, recognized by lymphocyte. Tadi kan B cell dengan T cell, sebab B cell and T cell has receptor for that specific antigen of a pathogen, elicits an immune response, stimulate or cause or activate immune response. And then maybe, so the antigen may be component of a pathogen, such as toxin. So the toxin secreted by the pathogen can be also considered as antigen. Okay, so that are released into the extracellular fluid. Okay, so the toxin of the pathogen can circulate anywhere in your body. So this toxin is also considered as antigen because it triggers your immune system against that pathogen. Okay, so proteins or the antigen can also be proteins or glycoproteins on the surface of pathogen, foods or pollen. So in this case, they akan trigger a condition, we call it as allergic reaction kan, sebab bukan semua orang akan ada reaction to certain food or reaction to certain pollens kan. So orang yang sensitif, orang yang ada allergic, so dia akan also trigger an immune response. Okay, which is allergic reaction. Okay, such as on the surface of transplant and also on the surface of transplanted organ kan. So transplanted organ is also considered as foreign to your body. Okay, so sometimes transplanted organ can be rejected by the recipient body. Okay, so bacteria kan, so virus, obviously they are pathogen, they are foreign to your body, so it can trigger your immune system to respond and attack this pathogen. Okay, so the next molecule or the next term that you have to know is antibody. Okay, so it is also known as immunoglobulin or Ig. Okay, it is a protein secreted by the B cell. Okay, secreted by the B cell or the plasma cell. Okay, plasma cell secreted by the effector B cell. Boleh tambah kat sini. Okay, effector B cell or plasma cell. So has antigen binding site that is complementary with the antigenic determinant or epitope found on antigen. Okay, so binds to an intact antigen. Okay, and then interferes with the pathogen activity. You have to remember for this one, the function of the antibody is to interfere with the pathogen activity. Antibody does not destroy the pathogen directly. Okay, the antibody does not destroy the pathogen directly. It will interfere with the pathogen activity, such as, for example, it can cause the antigen to clump together or agglutinate, or it can cause the antibody of that pathogen or the pathogen to clump together, and then once it is clumped together, it is easier for macrophage to come and then engulf. Okay, so the antibody, remember, it does not destroy the pathogen directly. Alright, nanti kita akan tengok ada cara dia macam mana antibody terlibat dalam your immune system. Okay, so how the antibody works. Okay. Okay, so as you can see, so in this diagram, as you can see, this is an antigen molecule. Okay, so this antigen molecule is from a certain pathogen. Okay, a certain pathogen or pollen or food that is outside of your body. It is foreign. So it also has several epitopes. In this case, ada 3 epitopes. So what is the benefit of binding to the epitope? So that for one antigen molecule, you can have several antibodies, several different antibodies that are possible to bind to the antigen. Kan? So instead of kalau dia ada satu je shape, satu je lah antibody, tapi hikmahnya adalah kalau dia bind kepada kepada epitope. An antigen molecule can have several epitopes, so several antibodies can detect that antigen. Okay, several antibodies can detect that antigen. Okay, so maksudnya satu virus can have several different antibodies against that pathogen. Okay, due to the binding to the epitope region. Okay, so an antigen molecule can have several different epitopes that binds to different antibodies where it matches with the shape of the antigen binding site, okay, of the antibody. Okay, so this is the shape of an antibody. So as I said, antibody has the same shape as the B cell receptor. Same shape as the B cell receptor, except that it does not have the transmembrane region. Okay, because antibody is secreted into your body fluid. So yang lain tu dia ada sama je. Antibody has two heavy chain, okay, has two light chain, and then you have the constant region, you have the variable region. The variable region will determine the antigen binding site, okay, and then all these chains are connected by disulfide bridge, okay, except that antibody does not have any transmembrane region because it is secreted into your body fluid, okay, into body fluid, okay, blood, interstitial fluid, and also lymph, okay. Okay, so now we are going to look at antigen presenting cell, or APC. Okay, APC. So as I said, there are three possible cells that will become APC: macrophage, dendritic cells, and also B cells. So these are cells that will engulf or internalize, okay, the Pentogen. Okay, so and then after the Pentogen is being internalized or engulfed, so this Pentogen will be destroyed, okay, or degraded into fragments, okay, into antigen fragments. So these three cells, macrophage, dendritic cells, B cell, function as APC that displays foreign antigen and their own surface protein. Surface protein here is referring to MHC molecule. Okay, MHC molecule. Specifically class 2 lah, sebab dia melibatkan APC. APC akan present antigen fragment menggunakan MHC class 2. Okay, menggunakan MHC class 2, dia present kepada siapa? Kepada helper T cell ataupun T helper cell. Alright, so APC originally they are inactive. Okay, they are inactive until their receptors recognize a particular molecular structure on pathogen, and then when activated, the APC ingested the Pentogen. Okay, ingested the Pentogen. Macam saya tak sure ada tak gambar. Sini internalize lah, engulf, ataupun ini by macrophage. Kalau B cell, dalam gambar ni dia tak ada. Kalau tengok, ini B cell kan. So once the B cell binds to the specific pathogen, dia juga boleh engulf, masukkan ke dalam. Melalui proses apa? Dulu kita belajar kan, receptor mediated endocytosis. So dia B cell pun akan boleh bawa masuk. Okay, and then becomes APC juga. Okay. Alright. So APC ingest the pathogen, internalize and ingest, degrade the pathogen using what? using lysosomal enzyme. Okay, so bila dia dah bawa masuk, kan, this is the virus, bawa masuk, masuk dalam vesicle. Okay, masuk dalam vesicle. It is packaged into vesicle, and then you have another organelle inside this macrophage or APC, which is lysosome. Okay, lysosome. So lysosome containing lysosomal enzyme will fuse with the vesicle containing the virus, and then degrade the virus or degrade the Pentogen. Okay, degrade. Okay, degrade the Pentogen or the bacteria into fragments. Okay, into fragments. We call it as antigen, antigen fragment. So this APC will display the foreign antigen fragments on its cell surface with a type of self molecule. We call it as class 2 MHC. Okay, class 2 MHC is a protein molecule produced by the APC. Fungsi dia apa? Untuk present the antigen fragment on the surface of the APC molecule. So nanti kita tengok gambar rajah dia. So the activated APC such as macrophage releases a signaling molecule. This signaling molecule can be called as cytokines. Okay, cytokines. So signaling molecules along with the displayed antigen and present displayed antigen to helper T cell. Dia nak trigger siapa? Dia nak attract siapa? Dia nak attract helper T cell. Okay. So if it involves dendritic cells, so dendritic cell also can become APC, and then it leaves the epithelial lining and guided by chemokines. Okay, migrate to the lymph nodes where they activate specific T cell capable of responding to antigen. So kalau kita tengok ni, kan, so this is a macrophage, for example. So the macrophage will engulf the Pentogen and then internalize and then fuse with the lysosome, degrade the Pentogen into antigen fragments. So nanti akan ada yang molekul hijau ni. Molekul hijau ni adalah class 2 MHC molecule. So this class 2 MHC molecule will bind to the antigen fragment and goes to the cell surface. Presented the antigen fragment, okay, to the helper T cell. This helper T cell will have receptor. Okay, so the helper T cell with the receptor matching with the antigen will bind. Only specific T cell that matches, that has receptor matches to the presented antigen will respond lah. Okay, so once you have this interaction, this macrophage, now we can call it as APC lah. Once they present, it becomes an APC molecule. Okay, APC molecule, dia akan present kepada helper T cell. So once you have this interaction, the APC molecule will secrete a signaling molecule. We call it as cytokines. Okay, cytokines. So you have another type of major histocompatibility complex atau MHC, which is produced by infected body cells. Okay, infected body cells. So sel-sel lain awak ni dalam badan yang ada nukleus, okay, when it is infected by the virus masuk dalam sel badan awak. So the virus will be fragmentized into fragments, into antigens, and then your body cell will produce class 1 MHC. Okay, so the antigen fragment will bind to the class 1 and then goes to the cell surface, present the antigen molecule to cytotoxic T cell. Okay, present the antigen fragment to a specific cytotoxic T cell that has receptor that matches with the antigen presented using class 1. Okay, so okay, so the MHC complex as I explained, kan. So MHC, major histocompatibility complex, it is a protein molecule. Okay, protein molecule encoded by group of genes. Ini dah belajar dulu kan, kalau untuk hasilkan protein mesti daripada gene dalam apa punya DNA tu. So daripada gene baru menghasilkan mRNA, through transcription, translation, hasilkan protein, which is the MHC. So what is the function of the MHC? It is to distinguish, okay? It enables your immune system to distinguish between self-antigen and also non-self-antigen. Okay, self-antigen and non-self-antigen. So it binds to antigen fragment and displays the fragment on the host cell surface, which is antigen presentation. Kan? So kalau dia adalah macrophage, dendritic ataupun B cell, so dia akan jadi APC lah. Tapi kalau yang dia akan attract helper T cell. Tapi kalau dia adalah nucleated cell, okay, dia akan presentkan kepada cytotoxic T cell. Okay. So ingat kita ada dua jenis MHC, class 1 and class 2 MHC. So class 1 found on all nucleated cell, class 2 found on B cell, macrophage and dendritic cell that will become APC. Okay, so class 1 will present antigen fragment to the cytotoxic T cell so that the cytotoxic T cell would destroy the infected body cell. Okay, and then for the class 2, it is presented by APC. It is to attract helper T cell. Okay, so this one I already explained to you. Okay, maybe this is the last slide before we end. Okay, so self recognition by MHC molecule, so act as self marker. This is the function of MHC. Dia bertindak sebagai self marker, the host marker. Okay, the host. So act as self marker to enable the immune system of the host to distinguish self from non-self. Okay. So the concept of self and non-self, so the mature B and T cells can differentiate self-antigen and non-self-antigen. So your immune cell, which is the B cell and the T cell, should be able to know which is self-antigen, which is non-self-antigen. So self-antigen is found on host cell and does not trigger an immune response. Okay, so it means that the B cell and T cell will not react to the self-antigen. If your immune cell react to the self-antigen, it can cause a disease. We call it as autoimmune disease. Okay, disease. So kalau non-self-antigen, it is found on foreign cell that enters the body and triggers an immune response. Yang foreign, this is the one that your immune system supposed to attack. Okay, the non-self-antigen, the foreign molecule that enters into your body. Okay. Okay, so I guess we can stop to this slide. So do you have any question regarding what we have covered today?